Did you know that yes, you, are made of many cells that are combined together? Not only yourself, but every living matter in this world.
That's right, Naufal. In this video, we will learn about cells. But before we do that, Arva, don't you think we should introduce ourselves first?
Yep, we should. After all, this is our first video too.
Yeah. You are right.
I'll go first. Hello, my name is Arva Muhammad Raditya. Call me Arva.
Hi, I'm Putri Kurotain. You guys can call me Rara.
Yahoo, my name is Muhammad Naufal Hafiz. Please call me Naufal.
And we are from class 10-4 in MH Thamrin High School. We hope that you will stick until the end of this video.
Before we are going to explain about cell structure, I'm gonna tell you a little bit about cells. As I said before, a cell is the simplest unit that makes all living organism. Mainly, cells are divided into two main categories. First is eukaryotic cell, and the other one is prokaryotic cell.
Thanks, Naufal, for the information. Alright, I'm gonna start explaining about cell structure. This is a eukaryotic cell. More specifically, this is an animal cell. As you can see in that picture, I marked something right. That thing is called cell membrane. Eukaryotes and prokaryotes both have a cell membrane or plasma membrane. It formed from phospholipid bilayer. The function of a cell membrane is to control the exchange of materials between the cell and its environment.
Next, we have nucleus. A fun fact about nucleus is it can only be found in eukaryotic cells. Nucleus have a function to store genetic information. Inside the nucleus, there is a region called nucleolus. The function of it is to produce ribosomes. Also, the nut cell of nucleus has the role to control the cell.
Next one is mitochondria. Mitochondria is an important structure because it holds an important function in the cell. It's to produce ATP or energy. Mitochondria is also defined as a prokaryotic cell that lives inside a eukaryotic cell. Because mitochondria have its own DNA and have a 70 Svedberg's ribosome like a prokaryotic cell. And this structure can only be found in eukaryotic cells.
Eukaryotic cells also have a structure that prokaryotic cells don't have. It's called Golgi apparatus. Golgi apparatus have a function to collect and process molecules within the cell. Golgi body will receive vesicles from rough ER and modify the protein by adding lipid and carbs, and then Golgi body will export the protein out of the cell.
Another cell that I want to explain is endoplasmic reticulum. It has two types. First is rock ER. It contains a ribosome, and because of that, rock ER have a function to produce proteins. Rock ER also can transport the proteins using vesicles that will be processed by Golgi apparatus. The second one is smooth ER. It synthesizes lipids. The difference between rock ER and the smooth ER is that rock ER contains a ribosome, while the smooth ER doesn't. Smooth ER, raw ER, and Golgi apparatus work together to produce modified proteins by combining the lipids, carbs, and protein that produced by rock ER.
Another cell structure that can only be found in eukaryotic cell is lysosome. Lysosome contain digestive enzymes called hydrolysis because they carry out the hydrolysis reaction. These enzymes need to be kept separate from the rest of the cell because it can digest the whole cell. Lysosomes have a function to get rid of the unwanted cell components like a broken cell component. Lysosomes also have a function to do a self-digestion. For example, if the cell are infected with cancer cell, lysosome will release all of its content to the cytoplasm and digest the cell.
Next, we have cytoplasm. Cytoplasm is basically a fluid like a jelly that contains all the living material outside the nucleus, like free-floating ribosomes, vesicles, etc. If you didn't know, cytoplasm can be found in both eukaryotic cells and prokaryotic cells.
Another cell structure that exists in both main categories is microtubules. It's a long, rigid, hollow tube that is found in the cytoplasm. They are very small, about 25 nanometer in diameter. With microfilament and intermediate filament, they make up the cytoskeleton. Microtubules made up of tubulin and have a function for structural support and transportation.
Next, we have fat wall. Fat wall can be found both in animal cells and plant cells. Animal cells are relatively small and include phagocytic vacuoles, food vacuoles, and autophagic vacuoles, while plant cells have a large central vacuole. Vacuoles are present in prokaryotes, but they are considerably smaller in comparison to eukaryotic vacuoles. The function is as a storage of food or a waste product.
In eukaryotic cells only, we can also find centriole in animal cells only. Centrioles lie close together and at right angles to each other region known as the centrosome. Centriole is a hollow cylinder about 500 nanometer long, formed from a ring of short microtubules. The function of centriole is to help split the cell apart during cell division.
Next, we have ribosomes. Ribosomes are very small and not visible with a light microscope. Size of ribosomes often quoted in S units. Eukaryotic ribosomes are 80 Svedbergs and prokaryotic ribosomes are 70 Svedbergs. The function is involved in protein synthesis.
Let's change the picture of an animal cell into a picture of a plant cell. Only plant cells and prokaryotic cells have a cell wall. Cell wall is formed outside of the cell membrane and has the function to provide structural support to the cell. There is a structure called plasmodesmata. The function is to connect the cytoplasm of neighboring cells. In eukaryotic cells, we can find chloroplast, but we cannot find it in prokaryotic cells. Like mitochondria, chloroplast is surrounded by two membranes which form the chloroplast envelope. Chloroplasts tend to have an elongated shape and a diameter of about 3 until 10 micrometer. The main function of chloroplast is to carry out photosynthesis.
That's all for the eukaryotic cell structure. Now we are going to learn about the cell structure in prokaryotic cells. Some of the structure have been mentioned before, so we are just going to explain about the structure that can only be found in prokaryotic cells. Let's take a look at the picture. The first structure is cilia and flagella. They are also play a role on the movement of the cell. Cilia and flagella are extremely complicated structure, composed of over 600 different polypeptides. Cilia have two central microtubules and a ring of 9 microtubule doublets around the outside. Cilia consist of tiny short hair-like structure and that's many of them, and allow the cell to stick to its environment. Flagella consist of long whip-like structures and there is only a few of them. Flagella can propel a cell forward from one location to other.
The second one is capsule. It's an outer protective covering found in bacterial cells in addition to the cell wall. It helps in moisture retention, protects the cell when engulfed, and helps in attachment of cells to nutrients and surfaces.
And finally, we have plasmids. Plasmids are non-chromosomal DNA structure. These are not involved in reproduction. And that's the end of the cell structure explanation in both eukaryote and prokaryote cells. After this, we're going to explain about endosymbiosis theory.
Endosymbiosis theory. There was variety in prokaryotes. There were photosynthetic prokaryotes, and some of these prokaryotes were bacteria that had the ability to use oxygen to produce ATP. Some of the prokaryotes were large and could engulf some of these small prokaryotes. But instead of the small prokaryotes getting digested, some of them remain intact and they live together.
This is believed to be the ancestor of heterotrophic eukaryotes. Eventually, some of these cells engulfed the small prokaryotes that could do photosynthesis, and there was more living as a symbiont. This is believed to be the ancestor autotrophic eukaryote. Endosymbiosis isn't just reserved for an ancient theory that explains a passive in ancient, but also happens today with many other kinds of organisms.
The example of endosymbiosis is the termite. Termites can have prokaryotes that live in their gut and help them digest wood. Host cell and prokaryotic cell experience symbiosis, where prokaryotic cells transfer the DNA to host cell nucleus. That's all about this video. I hope you enjoy and understand all of the information we gave.
Pardon us if we had a mistake in pronunciation and grammar.
Thank you and see you next time.